that. And then John Butler, you're up. So John
Okay.
Butler, co-founder and CEO of Quantumcyte. Take it away, John.
Thank you very much. And Uli and the MD Innovator staff, thank you for giving me the opportunity to present. It's kind of an update. I think I presented 2 years ago, and you'll see the progress we've made. So again, my name is John Butler. I am the CEO and founder of Quantumcyte. We're a Sunnyvale-based health tech company, early stage health tech company. And what we've done is we've developed an AI integrated tissue dissection solution for the modern pathology clinical markets. Our tagline is we get answers from every biopsy. So I wanted to start off with introducing us because I think we're a little bit different company than companies that are presenting. We're a tech company. We build technologies. What we do is we work with MDs, we work with oncologists to get requirements for unmet needs or issues that they're facing in their labs, and we develop technology to solve that problem. So again, I'm the CEO and CSO of the company. I've been working for about 30 years in the biotech industry in the Bay Area, developing technologies to accelerate biology. And Dr. Badant Chaudhuri, my CTO, and COO and co-founder, he comes from the high-tech industry. And it's important because what we have developed is we've actually developed a biotech solution at high-tech scale, and that'll become apparent as I go through the talk. And this is our SAB. Really love our SAB. They're very responsive and great group to work with. So where the company sits, we sit between, or we straddle the digital pathology world, either AI or manual digital pathology systems and molecular analysis. So we bridge the gap. We can take the digital information and use that to go and create clinical workflows or workflows to do molecular pathology. So our value proposition is about 25% of cancer patients' solid tumor biopsies that are submitted for a given clinical next generation sequencing based test. Those patients do not receive conclusive results. This results in delayed care for the patients, fewer options for care, as well as a negative impact on the healthcare system, as well as lost revenue for labs. And the reason for this has to do with a step in the process for harvesting tumor from that biopsy. The current workflow uses low image resolution or manual marking to identify where the tumor is, and then a technician goes in with a razor blade and tries to harvest tumor to extract nucleic acid for the downstream nucleic acid analysis. This is fine for the 75% of the samples. It's not adequate for the 25%. What we've done is we've created this high-tech solution where we integrate high image resolution digital annotation along with our automated tissue dissection system to effectively increase the purity of the sample of the DNA to meet a specification that's required for good NGS data. That specification is greater than 30% DNA purity or nucleic acid purity. We don't touch what the workflows that the pathologists do, and we don't touch the downstream nucleic acid analysis. We are a pre-analytical step. And so the way the system works is shown here. On the left-hand side, we've got an AI model that we built in-house using the iPhoria platform for heart disease. This was work we did in conjunction with the University of Heidelberg. I'm going to stop it there because you can see the regions that have been identified are the regions of interest that we want to extract nucleic acid from are here shown in yellow. What we then do is we take this digital image and we create a black and white image. Black are the areas that we want to exclude from extracting DNA or nucleic acid. We do not want the DNA from there. And the white areas are the areas that we want to extract nucleic acid from. And here's where the magic happens. What we've done is we've developed an ink that when we dispense directly on top of the tissue, it dispenses in the black area and it blocks those areas from the next step in the process, which is using chemistry, an enzyme to digest the cells in the open areas and the unmasked areas and release the nucleic acid into solution. So I'm going to come back over here. On the right-hand side is our instrument. You see two tissues, two biopsies on the deck, and you'll see that this inkjet printer actually moves and it does its job. We add the mask, and then using standard lysis reagents from kits that have already been qualified in clinical workflows, we extract nucleic acid from that region, the open region, and then we put that into a tube, and now you're free to do whatever nucleic acid prep and analysis you want to do. Here are some images of the mask and how the mask performs, and I'll be quick about this. If you look in the lower right-hand side, you'll see the mask, which is a serrated area. And up to the left, you have the tissue that has not been lysed, and to the right, you have the regions that have been lysed. You can see nuclei go right up to the mass. So we have this incredible precision and accuracy to go in and extract from those regions. And then if you want to look at a very complex tissue, this is a lung cancer sample we did in a collaboration with Singapore General Hospital, where the manual process would in no way be able to separate tumor from the immune infiltrate in this case. And we were able to successfully mask out this very complex tissue when we're waiting on the sequencing data. We'll get that next couple of weeks. So a case study we did with Singapore General Hospital was really done to demonstrate that we can increase sample purity from these samples that otherwise wouldn't pass the specification. There were 3 cases in this case, and we did more, but I'm just showing 3 cases for this particular study. And in this case, we use manual digital annotation using a Philips system where the pathologist annotated, sent us the digital images, sent us the slides of tissue. We ran it through our process, and then we shipped the crude lysate, in this case, back to the lab, and they put it into their clinical workflow to look at the results to see how it worked. And here are the results. If we just focus on column 3 and column 4, and we look at the quality score and the allele frequency, what we're looking for is an increase in the quality score and allele frequency to show that we did in fact get an increase in sample purity. In this case, we did, and we were successful. I also want to mention that we had the system installed in Singapore General Hospital, and we're currently running 2 clinical trials, one for lung cancer and one for colorectal cancer. And we'll have our very first clinical data next quarter, so we're very excited about that. In another study, I want to show, this is with a company called Ochre Bio. Here they needed to spatially sequence, in this case, 1,000 donor livers for NASH or fatty liver. They had to do this in 1 year. So they teamed up with us, and we teamed up with Reveal Biosciences. Reveal did the AI on this because the only way we could annotate 1,000 livers was with an automated system. These were core biopsies, and you can see the size of the regions of interest in blue were about 200 microns in diameter. You could not do the manual process on these tissues. We got the AI running. We ran our process, shipped the RNA to a company called Lexogen, and then Ochre Bio did the computational work. I'm happy to say that we got some very good results. We saw genes that we expected to see, and several novel genes. And Ochre is now analyzing this data and validating some of those potential targets. What's of significance is the turnaround time for us versus the existing technology on the market that could do this, would do the study, which is laser capture. We had about a 10-fold increase in the throughput. So we can do large studies very quickly. And one other study, just to briefly talk about, we're working with Mayo Clinic on a paid pilot to develop a clinical test for colorectal cancer. Here, we're going to integrate their current AI-based clinical test for digital pathology clinical algorithm with our system to develop a clinical workflow in the lab. And then the goal is to expand our system within Mayo's Rochester facility. If we compare ourselves to the other existing technologies on the market, there are 3 that can do tissue dissection. We're the only system that can do both the hard samples and the easy samples. And us, as well as the company Zyal, are integrating the digital path directly into the system. Laser capture and millisect are still taking the images, and they can't take it from the digital path or digital output and do the extraction directly. Our business model, we have a recurring revenue model that includes systems, reagents, software licensing, so we're selling an instrument and consumables into the labs. Some of the constraints that we see, that we have the instrument constraint has been resolved. We have an off-the-shelf CMARC system that we borrowed from repurposed from the solar industry. All of our consumables are off the shelf, and we're going to have to hire salespeople up to 30 by 2026 to hit our revenue targets. I mentioned the system was repurposed from the high-tech industry. Seuss Microtech supplies the system to us. There's a SKU, we can order it, we can get it in our lab, and get it to our customers in 3 months or less. And here's a system that's actually in assembly and final test. Our total addressable market as calculated by us is about 3.5 billion in 2026. And here are some of the assumptions that we put into that model to get to these numbers. And our revenue model that we've developed, we see about $60 million in revenue in 2026. And some of the drivers include the US and Singapore markets, early access, which we are in Singapore and the US, and then the difficult cases and automation, meaning the other 75% of the cases will pick up, expansion into pharma, and then also expansion into Latin America and Asia Pacific. And we're already talking to people in Latin America to extend. This is a sampling of some of the customers that we are currently working with, including pharma and academic hospital systems. And this is, again, a snapshot of our sales pipeline. So the problem that we're resolving is the inconclusive biopsy test results. We resolve an important bottleneck in the clinical workflows, and our customers are clinical labs and hospitals, commercial labs selling clinical tests, as well as pharma. We do have a current raise with a term sheet for a $2.5 million raise with a $32 million post on a safe. We have 800, and I think earlier today, $1 million committed with $1 million to $2 million in soft commits. Those funds will be used to fill the sales pipeline, increase our capacity, service 3 projects, Singapore General Hospital, Mayo, and another biotech company, and then to increase our sales and marketing outreach to validate our revenue models. I'd like to thank you for your time.
Impressive. Thank you so much. This is very the last minute up to date technology. Amazing. Yuli, you wanna take it away?
Good. Are there any questions so far for our presenters? Any last minute questions?
I do.
Yes.
This is Vipula here. A great presentation. Very impressed, John. Quick question. Are these algorithms that you guys have, is it only for oncology space, or can we do that for
No, we are tissue agnostic.
Okay.
We're focused on oncology. It's a market that we can define, but we did work in NASH, and the model that I showed you was for heart disease. So as long as it's a fixed formalin paraffin embedded tissue on a standard microscope slide, 5 microns, we can work on it. Yeah.
Okay. Can we connect offline? I'd like to get more information. Thanks.
Thank you, Vipula. By the way, Vipula is also an accomplished exec, but also an investor. So thank you for that. Any other questions?
So Yuli, I do want to apologize for going so quickly. I actually thought I had 15 minutes to speak.
No problem. We try to push people. Okay. Thank you very much. Looks like there are no more questions.
Thank you, Yuli.